Bacterial protein extraction. This process involves culturing of bacteria, harvesting and sonication of the obtained culture, followed by protein extraction. Bacterial culturing. Clean the laminar workspace thoroughly with ethanol and keep the master plate having the bacterial culture ready. Light the spirit lamp or Bunsen burner to maintain sterile and aseptic conditions throughout the process. Carefully remove a bacterial colony from the master plate and inoculate the autoclaved growth media. Incubate this inoculated sample at 37 degrees Celsius for 6 to 8 hours with constant shaking to allow the bacteria to grow. The turbidity of the culture, which gradually increases, indicates bacterial growth. Culture harvesting. Transfer the grown bacterial culture to a fresh tube under sterile conditions. Centrifuge this tube at 12,000 rpm for 10 minutes, maintaining a temperature of 4 degrees Celsius. Transfer the pellet obtained containing intact bacterial cells to a fresh microcentrifuge tube. Wash this pellet thoroughly with phosphate buffer to remove any unwanted debris. Sonication. The resuspended pellet is sonicated on ice to enable the bacterial cells to break open so that its contents are released. Sonication involves the use of high-energy sound waves that are capable of breaking open the outer membranes of cells. All cellular contents, including proteins of interest, leak out of this disrupted membrane. Carry out the sonication procedure for 30 seconds with a pulse of 1 second at 40% amplitude. Once it is complete, centrifuge the contents and collect the supernatant that is obtained. Protein extraction. The trizol extraction protocol allows efficient separation of not just the bacterial proteins but also their DNA and RNA. Add the trizol reagent consisting of quinidinium thiocyanate, phenol and chloroform to the supernatant obtained after sonication. Mix the contents thoroughly by vortexing. Next, add chloroform to this solution. Mix the contents and place the tube on ice for a few minutes. Centrifuge the tube at 2000 rpm for 5 minutes. 3 distinct layers will be obtained. The topmost is the aqueous layer containing RNA, the center is the interface containing proteins, and the bottom layer is organic and consists of DNA. Discard the transparent top layer having the RNA. Then, add absolute alcohol to the remaining layers and mix the solution well. Centrifuge the contents at 2000 rpm for 5 minutes. The DNA forms a white precipitate at the bottom of the tube while the proteins remain in the clear supernatant. Collect the supernatant in a fresh tube. Then, add chilled acetone to this tube and mix well by vortexing. Store this solution at -20°C for at least an hour before centrifuging it to obtain the protein pellet. Discard the supernatant and dry the pellet at room temperature. Reconstitute the dried pellet with rehydration buffer and store overnight at -20°C before carrying out protein quantification.